I will talk to you today about one project that we are leading here at the center called Polyethnic-1000. And the subtitle is Advancing Cancer Genomics by Studying Ethnically Diverse Underserved Patient Population in New York. So I'll start with those differences that we can observe. And so we go at the New York State Cancer Registry. But you see here, if we divide the patients by their ethnicity, we observe already some difference in the incidence of cancer between the non-Hispanic white, non-Hispanic black, in blue are the Asians, and in green here are the Hispanic. That's both for male and for female. We observe differences between those groups. If we go to colon cancer, we observe, again, some differences that are similar, although a little bit different. We see that we have more female non-Hispanic black that have colon cancer than white female. If we go at breast cancer, here is the statistics for incidence. Now, if we go at mortality, we also observe similar difference for all cancer. Those differences are similar to the differences in incidence. If we go directly to breast cancer, we see here that non-Hispanic black women have a more lethal form of breast cancer. So they have a little bit less incidence of breast cancer than white women, but it's more lethal. Okay, so there are some differences. And obviously, there are multiple sources of those differences. The first one we can think of is what we call socioeconomical factors. So healthcare access, lifestyle, risk behavior, diet, all of those things can play a role in incidence occasionally, but also in mortality, in the way the cancer develops. Those things are very important. They can be studied. There are a lot of people collecting large amount of statistics, large amount of data, trying to tease out the different sources of those disparities. What we're mostly interested in are the molecular differences between ethnicity and the way the cancer develops. So I'm going to show you two examples. This one is the frequency of the pathogenic variant in breast cancer or ovarian cancer in white population. And you don't see really the name of the gene here, but you can observe this one called CHEK2. And you see that it's present in maybe 25% of this cohort of white women. It's frequent at a similar frequency in Ashkenazi Jews, but it's much less frequent in Asians and in Hispanic, where the proportion of BRCA1 mutants is higher than in the white. Okay, so what are the genes that we know are associated with the development of cancer? We see that their frequency is different in different populations. And this is another example, an even more striking example, and that's the somatic variant, the variant that are only in the tumor for lung cancer. And one very common gene that is frequently mutated in lung cancer is this growth factor called EGFR. We know that it's mutated in about 50% of Asian patients, and in between 5 and 15% in European population. So there's a very common gene. It's a gene that's very frequently mutated in Asians, much less frequently mutated in European. We know it's not associated to diet, to environment. We don't really know what's the source of this major difference, but we know there are more of them, and that's what we want to study. So I prepared a few slides really to go very quickly on what we do at the Genome Center when we do cancer genomics. We take normal cells, we take tumor cells, we extract the DNA. We know that there are a lot of differences between in the tumor DNA. We chop those DNA molecules into small fragments. We sequence them on an Illumina sequencer. We sequence about 150 nucleotides for each of the fragments. And then we look at their position in the reference genome. We do this process called alignment, and that's represented here with a color for each of the nucleotide. You don't see here, but there is one difference between the normal alignment and the tumor alignment here. And that difference here is a hotspot mutation in a gene called KRAS. So that's mutated in about 75% of pancreatic cancer, for instance. So what we have to do is differentiate between sequencing errors. So our sequencers are extremely powerful, extremely good, but occasionally, randomly, they make an error. And because we generate billions of reads, we can have some occasion mistakes. So we have to remove or ignore them. We also have to identify variants that are found in both the normal and the tumor, and those are germline variants that we will capture in both experiments, and focus on the variants that are only found in the tumor and that are represented here. This is a cartoon example for a simple form of variant, just the single nucleotide substitution, but we also have many more complex events that are summarized here. We have multiple strategies to identify them. Those are the ones that lead to the very messed up karyotype that Simon showed you before. So just for the bioinformaticians in the room, this is the description of our pipeline. We take the reads, we align them to the genome. We use different variant colors to identify all of the different sort of variants that are unique to the tumor that are not found in the germline variant. I need to highlight here that two of the tools that we used were developed at the New York Genome Center, one by my colleague Giuseppe Narcisi, who's another staff scientist in the computational biology group, and one by Marcin Emilienski, who's a faculty member at Weill Cornell School of Medicine and at the New York Genome Center. Another introductory slide on the different type of experiment we can do in cancer genomics. We only do we only do whole genome, whole exome, and RNA-seq here at the Genome Center, but the difference is the following. Whole genome sequencing is essentially trying to sequence from telomere to telomere of all of the chromosome in a particular sample. Whole exome sequencing is using a method to capture the region of the genome corresponding to protein coding genes. And that corresponds to 22,000 genes and about 30 to 50 million nucleotide. And panels can be variable size, about 1 megabase, can be from, let's say 300 to 500 genes, and there really will be regions of interest, genes of interest. And in cancer, we can select genes that we know are associated to cancer, and we can probe those genes only, sequence them, and so really focus on those ones. And then RNA-seq is the sequencing of the transcriptome. We sequence the RNA molecule, and that tells us something about the expression of the genes, and also some aberrant molecules that are only expressed in tumors. And the goal of all that is to identify recurrent patterns, driver mutations, so the mutation that really originate at the very beginning of the tumor and explain the proliferative advantage of those cells. And we can also try to identify targetable mutations, so small mutation that we can target with a drug and that will make the tumor respond to a treatment. We can also study tumor formation, tumor evolution, response, and resistance to treatment. Now, to go back to my question about health disparity, to my problem about health disparity, the main problem we have is that we have been doing cancer genomics for about a decade. We have accumulated a large amount of data through whole genome sequencing, whole exome sequencing, panel sequencing at various places in New York, in the rest of the country, in the rest of the world. And so we have a good idea of the somatic landscape of the most common cancer type. However, the main problem we have is that those data were obtained mostly from patients of European ancestry. So this is a summary here of the TCGA, the Cancer Genome Atlas, where about 75 to 80% of the samples are coming from patients of European ancestry. There are many reasons for that, but one of them is that it's being done at research center, and those research center primarily their patient population is heavily skewed. So that's the results of a lot of healthcare disparity in this country, but that's really true elsewhere as well, and that's a major problem for our field. So we have a good idea of what are the genes that are mutated in the cancer of white people. And as I showed you, we know about some cases where those frequencies are different in other populations. So we established this program called Polyethnic-1000 to try to study the variability of the somatic profile, try to identify, is this cancer type always behaving the same, always having the same type of mutation, or is that different between ethnicity? The goal is to have more people benefiting from genomic research cancer testing, including in the underserved population. And by doing that, we will establish a network of institutions in all of the neighborhoods of the New York area. So the reason to do that in New York, obviously, is that we have the most diverse population on Earth. We do that in 3 phases. The first one is a retrospective phase with whole exome and RNA-seq. And that phase is completed, and I will show you the first results. The second phase is a prospective phase where we will enroll patients, and we will provide clinical confirmation of the variants for a subset of the patients. And in the third phase, we will have targeted projects to test specific hypotheses. And so we have already collected ideas about particular cancer type and particular ethnicity where we think there might be a difference. And we will use our network to collect samples that will address those particular hypotheses. So this is the map of New York, colored by the census group. And you see in green are the white individuals or the white neighborhood. In blue are the Black or African American neighborhood. In orange are the Hispanic, and in red here are Asian neighborhood. Okay, so you can see Chinatown, you can see Flushing, you can see Brooklyn, you can see all of the diversity in New York represented here at a very, very high scale level, which is the census categories that we rely on. And so at the moment, we can do studies relying on those census categories because people come at the hospital and check a box. Are you identifying yourself as Black, Asians, Hispanic, or white? And so we have established a network of the people that the institution we work with, a lot of the research center that are part of the New York Genome Center, but also a lot of hospitals that are serving the population in the different neighborhood in New York. The first phase, the first results I can show you are represented here, where we have 92 samples. We are doing whole exome sequencing and RNASEQ, and we do somatic variant detection, so all the single nucleotide variant, the indels, the copy number variant. But importantly, what we do is genetic ancestry inference from the exome data. And so it's really a similar method as what 23andMe is using. We identify in our exome the variants that are what we call ancestry-informed markers that associate with a particular population, and we can estimate the proportion of those population in our samples. So the first track here is the self-declaration. You see on the left, the patients that are Black or African American. You see a large group here of Asian patients. And the lower track is our genetic ancestry estimation. And you see that you have a variable amount of genome that comes from an African continental population. And you see that for people who self-declare as Black or African American, they can have a contributing gene or African genome that goes from 100% to something like 20%, right? And so you have already a large disparity, a large diversity here, variability in the contribution of the African genome. You see here that Asians can be easily separated in East Asians and South Asians, okay? So those are the label here of what we call those suprapopulation or continental population. And that's already where we can see some diversity. But it's even more interesting when we go a little bit lower. We use now 25 reference population from a consortium called the 1000 Genome Project. And we can estimate the contribution of each of those reference population to each of the patients that we have here. And you see that in our African, or in our patients that self-identify as Black or African American, you have really a large diversity of African reference population that contributed to their genome. And that's really a level of variability that we think will be extremely important. And as we know, there is actually more genetic variability within Africa than between Africans and European or Asians, okay? And so that's here a level of variability where we think we might observe very important differences, for instance, in breast cancer between breast cancer in patients that have a huge contribution from West Africa or East Africa, or that level of differences. You see here that our Asian group here, we can identify the ones that are from Korea, the ones from Japan, from Vietnam, and so on and so forth. So we represent all our data into a CBio portal, so an interface that give access to the scientists to all of the data. This portal was developed mostly at Memorial Sloan Kettering, but we're using it for our project. They can start with the entire project, and then they can go deeper at the different sample, identify all of the variants for that particular sample, and get access to a lot of annotations about those variants to identify if there are some variants that are associated to a drug or to a treatment or to a clinical trial. So the first phase is represented here. It's a retrospective phase, tumor-only sequencing, and the patients were not consented, so this data is not going to be shared. But we are starting now a second phase that is prospective, where we do tumor and normal sequencing. The patients will be consented, and we will provide some clinical validation that will benefit some of the patients for cases where we will identify an actionable mutation that could change their treatment. And in the third phase, we will do targeted project. And at that point, we may have, for certain projects, the ability to change the type of experiment we do, not only do whole exome sequencing, but do whole genome sequencing, possibly collect some sample to grow cell lines, to grow organoids, to do single cell project, and so on and so forth. So all of the samples from diverse ethnicity will be deposited in public repository. There will be clinical benefit for a subset of the patients as early as the phase 2. And we will have a strong network of partnering institutions a workflow of the sample from all of the hospitals in New York to the New York Genome Center and a community of physicians and scientists to analyze this data. And in phase 3, we will have a specific research proposal that will leverage this network and this ability to collect, to enroll patients from a diverse population. And that can be expanded to whole genome sequencing, to clinical sequencing, to larger geographical area, and to enroll additional partners. So just in conclusion, we can sequence tumors, and we have already learned a lot through sequencing tumors. What we need now is to explore the variability of these tumors in diverse population, so that we can learn what we can generalize to the entire population. And really, from an artificial intelligence point of view, you can imagine that we generate very, very large database, and you can have maybe a naive view that if you have all the data and you have the outcome, you can train a classifier and learn all of the features from that database that will predict what is the outcome of the data. I think we're really far from there, so don't get too excited. At the moment, what we want is to have all of the prior information. The way a physician looks at a particular genomic report is to say, okay, I identify all of these variants, and based on my current knowledge about this cancer type, I will do this or that recommendation. I will give out this or that drug, or I will enroll these patients into this trial. As part of this prior knowledge, we need to incorporate the diversity that we will explore here. And so we need to know if the doctor need to know about the ethnicity, need to know about the will that change the prior information that will then change the treatment? And so if ever one day we arrive to a point where it's completely automated and there is a machine learning or an artificial intelligence that does this process, we need to know if what it can learn, or in order for what it learns to be applicable to the general population, we need to have the diversity built into the data that it learns from, so if that makes sense. I can get back to this point after. But so we're setting this consortium to enroll patients from ethnic minorities in New York, to diversify the public repository, to identify potential differences in tumor profile between ethnicity, and to make sure that every patient will benefit from the advance of precision medicine. This is a representation of the people that work on this project, the Polyethnic-1000, the people that work with me, and the cancer genomics at New York Genome Center, all of the working group that help us set up this project, and the steering committee led by Hal Varmus from Weill Cornell, Charles Hoyers from MSKCC, and Dave Tuveson from Cold Spring Harbor. The funding for the pilot phase was provided by the Mark Foundation for Cancer Research, the Mortimer Zuckerman Family Fund, and Wesley Janaway from the Board of Directors of New York Genome. Thank you very much.